turns out that horizontal gene transfer, or HGT, you could also say LGT for lateral gene transfer. Turns out to be hugely important for the generation of new functions in a given lineage of microorganisms. And so in this case, what you've got is DNA, usually DNA, because these organisms have DNAs by and large as their genetic material, their hereditary material. You have a chunk of DNA that actually finds its way into a new cell and can be incorporated into that cell and thereby provide that cell with a new function. And we've actually got three general means by which traditionally we talk about acquisition of this new DNA. One of them is known as transformation. Now, and that involves the acquisition of naked DNA. That is DNA from the environment. It doesn't have any proteins associated with it. There's also conjugation. And this is the acquisition of a plasmid via the actions of what's known as conjugation or sex pili, where you have genes that are associated with a smaller piece of DNA that are transferred from one individual to another as part of the small piece of DNA. And then the small piece of DNA takes up residence in the recipient bacterium or organism. And any genes associated with it can be expressed and thereby modify the phenotype of the recipient organism. This is most important as far as we're concerned because of the potential for transfer of antibiotic resistance genes. So these plasmids can contain genes that encode for antibiotic resistance, and their transfer results in an organism suddenly, upon reception of these plasmids, suddenly being resistant to an antibiotic. And it gets even worse because often these plasmids, known as resistance plasmids or R plasmids, encode for multiple antibiotic resistance mechanisms because they have multiple antibiotic resistance genes on them. And therefore, a single bacterium can acquire resistance to multiple antibiotics simultaneously by simply acquiring a single plasmid. And then lastly, there's transduction. And transduction is mediated by what are known as bacteriophages, or at least in bacteria they are. So bacteriophages are viruses that infect bacteria. And what can happen is that a bacteriophage can inadvertently carry a piece of bacterial DNA from one bacterium to another, and thereby potentially carry new functions from one bacterium to another. In addition, a certain type of bacteriophage, known as a temperate phage, is able to go through what's known as a lysogenic cycle, and in doing so, the phage as a whole becomes incorporated into a recipient bacterium, and these temperate bacteriophages can carry on their genome, so we have a genome within a genome here, they can carry genes that can be useful, potentially useful to the bacterium they're infecting. So you've got a virus that carries genes on its own accord and is able to move from one bacterium to another in being acquired by the second bacterium. Those genes are now also acquired by that second bacterium. Those genes can be expressed and in the course of that expression change the phenotype of the bacterium. Ideally for the phage, for the virus, thus increasing the fitness of the bacterium, thus making it more able to interact with its environment and reproduce and survive. Okay, and this process of phages, bacteriophages carrying genes into new bacteria is known as lysogenic conversion. because what these bacteriophages are doing is they're creating what are known as lysogens by going through what are known as lysogenic cycles. And lysogens and lysogenic cycles is something that we'll talk about when we actually talk about viruses, and therefore talk about bacteriophages. And I kept using the word phage instead of bacteriophage because people often refer to bacteriophages instead simply as phages. And with that, I'm going to call it a day.